insert nucleus is more angular and has a very prominent nucleolus. I'm writing through totally thousands, that's a shorter word.
So, I guess.
Yeah, there is one, I just saw one, I got to back up a little bit.
Right
Here's
there,
one,
right there, yeah.
and here's one.
Is
No,
that one
these
up
are,
there?
yeah, they're just different styles. You can't tell the separation because they're so
Is that one
blended.
up there? Right,
Up here?
Right, that one, yeah.
Yeah, that's one up there.
How come it's not along the basal?
The nucleus? These are the nucleus of the sustentacular cell. So they can be anywhere inside the cytoplasm of the cell.
Oh, okay.
It's only the spermatogonia that are along the basal surface. Okay? Now, before we leave this view, we have another group of cells to indicate. And these are found outside seminiferous tubules in the interstitial space where the blood supply would be. These are the cells that synthesize testosterone. So these are identified as the interstitial cells. Of Leydig, or any combination of the above except cells. So Leydig cells, interstitial cells. Interstitial cells is rather vague, but it is often used. Interstitial cells just meaning the cells in the tissues, which is what every cell is. And these are found here.
Okay.
And here.
Testosterone.
They synthesize testosterone. So we'll talk about that mechanism Thursday and or Friday. So here's a lower power view of several cross sections, and the interstitial cells are found in the gaps between those cross sections, where the blood vessel, the capillary supply is located.
Pretty good size, right?
So on page 60, there's a description and an image of these. All right. If you have a histology atlas, such as Weider or LeBoff, there are similar images in here, okay, that you can take a look at. But again, for Thursday's quiz, just being able to identify it as the organ. All right, let's skip through the physiology part on page 62. We're going to come back to that. I want to give you some material to use in lab rather than just looking at physiology components. So this process by which the sperm undergoes these cellular and nuclear changes is known as spermatogenesis, the formation of new sperm. The process by which the late spermatids go through early spermatids and form the mature spermatozoa is known as spermiogenesis. Spermatospermia. So let me write those words on the board. Can I erase what's on the board? So the spermatozoa, or the early spermatism, lose a lot of cytoplasm. They basically become membrane vehicles for motility, transporting the nucleus. All the other material is extraneous and a load that the cell gets rid of. It basically takes with it the nucleus, mitochondria, and centrosomes, and a beating tail. So again, spermatogenesis is the nuclear changes, the chromosomal changes, going from spermatogonium to spermatozoon. Or early spermatids when we first see that changes. But then losing that cytoplasm and taking on this appearance is spermiogenesis. Developing into a sleep-named finding. Okay, so that, there aren't any genetic changes that occur here. It's just taking on this appearance. This is known as the head. The mid-piece. There's a neck in there, but. And the tail. Or flagella. Within the head, we have a nucleus, which is haploid. We've undergone meiosis. So because it's going to meet up with the egg, which is also haploid, then we're back to a 2N cell. All right? Pull down over the nucleus like a knitted cap is a membrane sac. Here it's in sagittal section, so it's really not like a U, it's like a full cap. Known as the acrosome or acrosomal cap. We'll talk about that again when we do fertilization. Because when the head of the sperm comes in contact with the egg, these two membranes disappear, and enzymes in the acrosome allow it to digest its way through the coat that's outside the egg itself. This develops or has its origin, so we talked about the cremaster muscle having its origin from internal oblique. The acrosome has its origin as one of the Golgi sacs. The other components of the Golgi degenerate, but one remains behind, known as the acrosome. The flagellum is like a very long cilium. Remember when cilia were cut in cross-section, they had 9 doublets? And these microtubules slid past each other using ATP enzymes to provide the energy, and that created motility. So that's what we see in the flagellum, and it has this kind of rotating, whipping motion. The enzymes aren't mature in the sperm until after they've been in the tail of the epididymis for a while. All right, but the structure is there. So the flagellum has these microtubules. Which start here in the mid-piece, kind of like the basal body. And wrapped around in a circular fashion, we have the organelles that are providing the energy. Mitochondria. Now there's not enough room in the sperm. Guys, if all of your spermatogonia and spermatocytes remain their original size, your scrotal sacs will be like this. Okay? So that only occurs if you have a parasitic infection now. But it'd be a lot of baggage to carry around, and a lot of volume in the ejaculate, and a lot of wasted components. So the egg keeps all of those, provides all of the those cellular organelles. And now the mitochondria needs a source of energy. So that's a major component of the secretions of the seminal vesicles that we'll talk about in just a moment. So here's a view of a sperm, human sperm, whose acrosomes have reacted, meaning the membranes have fused and been lost. males have, different animals have different shapes of heads. Human is kind of an angular arrowhead. A guinea pig, shaped like that. Yeah, maybe they're red. And a porcine, a regular pig, a hog, is shaped like a ping pong paddle. I don't know what advantage that provides in the attachment to the egg, but there must be some. So when I was I wasn't a graduate student, I was teaching at the med school, and I only had one child at the time, and he got to be 3 and started preschool. I was only teaching 2 days or 4 afternoons a week for their full-time position, so I had a bunch of extra time, and I volunteered in one of the labs. And they worked on sperm. And in fact, that lab was one of the ones to discover the requirement for progesterone in the in vitro fertilization of eggs to allow the sperm to undergo this acrosome reaction. So the lab worked with both human and pig sperm. I happened to use human. This was the early 90s, and we paid the donors, graduate students, and medical students about $25 a ejaculate. So when they take a little sterile urine specimen containers and bathroom right around the corner from the lab and bring them in, we had a CO2 incubator, and in the proper fluids and CO2 pH, we could keep them alive for about 2 weeks. Not the sperm, not the donors. And so they'd walk in with their ejaculate, and they'd see me sitting there at the desk, and I was their teacher for gross anatomy or histology, and they'd turn around and walk right out again. So I'd get up and go to the other lab room, and then they'd come back in and sneak it into the refrigerator. But I was working with human on electromicrograph, and I really needed a lot of sperm because I did a lot of washings. And the typical male ejaculate, and that includes the fluid from the glands, has a volume of about 2 to 5 mils. We include the sperm and all the secretions of the gland. Average number of sperm per mil is 20 to 25 million per mil. So at a maximum, you'd 20 to 25 to 50, sorry. So at a maximum, you'd be happy to get 250 to 350 million sperm total. That'd make a visible clump at the bottom of my little flask that I was renting out, graduated tube. My favorite donor, I have no idea what his name is, he didn't get any extra money. I got 1 billion sperm from him. And it wasn't like he ejaculated 10 mils, he just produced a lot of sperm. And so I could do whatever I wanted to do the experiment 20 times, and I still have sperm cells left over. But we would look at them under the microscope. They'd still be beating, make sure they didn't have 2 heads or 2 tails or some other malformation. And then I did try to keep them alive for 2 weeks. I would use them and right away attach gold particles, looking for different molecular receptors on them. The graduate student was working on hog sperm, graduated, got her doctorate, and the principal investigator, the PI, asked me to switch over to her research. Now remember, I'm doing this on a volunteer basis, okay? I have extra time from teaching. And so in considering that, I said no. I don't know how many of you are former ag majors or animal science majors, but pigs, hogs, cannot use electrical ejaculators like cows can. She had to do it herself. And then go home and take a shower before she came back to the lab. And I was like, you couldn't pay me to do that. I'm not doing it for free. So I stuck with my donor with 1 billion sperm that would just bring it to me and stick it in the incubator. Stayed with that.
Ugh.
So here's some sperm that are showing some various anomalies of the acrosome reaction collapse. Now there was an old theory that The entire human body existed in the sperm as a miniature. And here's proof. Okay?
Oh, God.
I don't know if that was photoshopped or some membrane abnormality, but it's kind of cute. Yes, when I was dating, I took my husband to the lab, and this is a different lab, but in the same building when I was working with Jacinta. I said, hey, you want to see your sperm? So I sent him to the same bathroom, and he brought back his ejaculate, and I made sure he could be a father to this, all his sperm were normal. He didn't know that was what I was checking for, but he just thought it was the coolest date he'd ever been on. All right, so
I have
if
you on.
I told that story 15 years ago, I would be blushing more than I am now.
It's
So my
on.
husband still can't believe the kind of stuff I tell you guys. My son either. My son when he was about 7, we had a neighbor that I didn't think he'd seen her in a few years because we'd moved and they'd moved. We were in the same area, but 4 or 5 miles apart. So he came to me one day and he said, Mom, how are babies named? And I said, well, honey, we've gone over this. Well, I said, you know about penises and the vagina and the sperm? And he said, yeah, I know that. He said, but is there any other way? And I said, yes. I said, people that can't make babies that way, well, take the sperm and mix it with the egg in a plastic bowl in the laboratory. And he goes, no, I mean, is there any other normal way besides the penis and the vagina? And I said, no, not really. And so he looks at me and goes, well, he said, I don't think Julia's going to like that very much. I said, like what? He said, the penis and the vagina part. And I said, I don't think so either. I need to talk to her mom. So I gave her a call and I said, do you know what our kids are talking about?
How old were they?
Oh, 7. So being an anatomist, my kids learn about this stuff from who knows what. I just didn't know her thought anything about it yet. So the next structure, as the sperm are compelled by fluid flow, hydrostatic forces, they move into the epididymis.